Venter looks at life differently than most of us. Where we might see the spark of life as something almost magical, Craig sees chemicals. A molecule called DNA that can turn inert material into something vital, something uniquely alive. Craig says he wants to use synthetic life to make the world a better place. He believes man-made microorganisms can help clean the environment and provide alternative fuels, for example. Summer 2003, Rockville, Maryland. Dr. Craig Venter and his colleagues have begun a project that few would ever dream of: the creation of a living microorganism in the lab. Now they must create man-made, self-sustaining, self-replicating bacteria. A living creature by any definition. He'll start with the techniques Ham and Clyde developed. By making a short piece of DNA with handles on each end, they hope it will hook up with a string of corresponding letters in the next segment. But after a year of long hours in the lab, Dan still can't get more than half the circle to link up at once.
DNA, as it gets larger, gets much harder to handle, much more brittle. There's not mechanisms for handling chromosomes out of cells.
Teammate Dr. Gwyn Benders has an idea that might push things along.
And then we're also going to try doing this in yeast.
Yeast, the microscopic organism that makes beer ferment and bread rise, has a remarkable innate ability to help DNA molecules link up. To assemble large pieces of DNA inside yeast cells.
The yeast itself, when it takes up the DNA, will combine the DNA at these ends to make bigger pieces.
Yeast recognizes these 4 overlapping Porter molecules and it assembles them into one complete chromosome.
In 2007, 2 years after they began, the Venter team now has the complete man-made genome for a bacterium. But they are not done. Because DNA in a test tube is just a molecule. It is not life. This is Craig Venter's explanation of what should happen during the transplant.
Cells are very dynamic processes. So we can draw a cell, and the chromosome in the cell is its software. And all the proteins made by that chromosome will define the structure and function of that cell. Now we take a new chromosome and insert it in that cell and replace the other one. We have whole new software. And now we have this new software that drives the production of only blue proteins. And what happens is the cells divide and we get 2 daughter cells. They only have the blue chromosome. The proteins are only from that blue chromosome, and they define a totally different species than we started with.
This is the moment Craig Venter envisioned 10 years ago. This man-made DNA molecule will be inserted into a bacterial cell that is also too small to see with the naked eye.
Have mycoides genome.
If Venter and his team have the genetic code right and make no mistakes in the transplant,
Let's hope it works.
a new synthetic species should come into existence. Microbiologist Li Ma uses a series of chemicals in precise amounts. The recipient cell membrane softens and it absorbs the new synthetic DNA. The synthetic DNA has been engineered to overcome the defenses of the recipient cell and to replace its natural DNA. If all goes according to plan, the man-made genome will take control and reproduce. This Monday is different.
So about 6 a.m. I came into the lab.
I was going to ask when you came in to...
And I was very nervous and shaking a lot. I just want to peek in here. It's like a colony.
The synthetic organism was engineered to turn new cells blue. The result is easy to see. A colony of new man-made cells. They have succeeded in making life. Craig is not the only one who believes there's an almost infinite number of future applications for synthetic biology.
So the challenge is going from these tiny microbes to billions of gallons.
Yeah, absolutely. That's the big gap.
I think we're going to see power sources allowed because we'll have ways to clean up oil spills. I think we're going to see the creation of microbes that can get in our bodies and act like vaccines and protect us against infection from things that are killing huge numbers of people around the world. They may even help us find ways to do space travel. They may be able to be put into our bodies in ways that would control the devastating effects of radiation on us. The sky's the limit on what you can do in the microbial world.
So this is our test facility for the algae fuel program with ExxonMobil.
Craig thinks his microbes will be able to take carbon dioxide out of the air and convert it into cheap, clean fuel.
I think this early 21st century biology will start to change the way that humans think about life and think about cells and think about our place on the planet.
It's a mix of feelings. That means we can do a lot of things. And that also means we have to be careful.
It's a giant philosophical change in how we view life, on understanding life at its basic level.
I think the headline, "Scientist Creates Life," is going to have a seismic impact on our culture. When you can sit there and say, I'm going to take base pairs of DNA and arrange them in some kind of a sequence that makes a bit of inorganic matter come alive, you're doing something that's unbelievably important in terms of our understanding that life boils down to biological processes, nothing magical, nothing ineffable, and something pretty scary.
The first revolution took place 10,000 years ago when humankind invented agriculture. The second revolution was the Industrial Revolution, in which we built machines and our technologies have become very powerful and sophisticated. I think this is the third major revolution of human civilization.
It's a bright line in history, and it's a scientific accomplishment. It's a philosophical accomplishment. Its meaning will be interpreted over possibly the rest of human existence because all the things that are potential after this stage. This will allow us to start to engineer things to change how we live on this planet.